Hi, in this video I'd like to show you how to build a virtual cell in Python from scratch. So let's start by answering the fundamental question, what even is a virtual cell? Or I do you one better, why is a virtual cell? But the motivation of building a virtual cell in the first place. So maybe let's illustrate it on two examples, because two most obvious use cases for a virtual cell, I think, are drug development and disease understanding. So let's use some fancy tool to illustrate it. And let's say that this is a healthy cell. This is a healthy cell, perfect. And now we have the same cell type, but in a disease state. So let's say that this is the same cell type, but we can see that it's quite dramatically different. It has a different color, different shape. And so we have, theoretically, these are two same cells, but in completely different states. So we may want to ask a question, okay, how do we take the diseased cell and make it healthy again? So this is exactly what we can do in the lab. Okay, we take a cell and we introduce, professionally called perturbations, would be basically poke it with different sticks, and we observe what effect it has in this state, in this cell. We can also ask another question, like, okay, so what do we need to do to take this cell from the disease state to a healthy state, but also on all of the other possible human cells, we don't want to mess them up. So how do we perturb this cell to make it healthy again, but also how do we introduce such a perturbation that also doesn't change all of the other cells in the human body? So like a targeted therapy. We can also ask a question. This is in particular relevant in cancer. What do we need to do to this cell to make it die, but for this cell and all of the other cells in the human body to live? This is in particular, as I said, important in cancer because this is the basis of many targeted therapies. There is also another question we can ask. And this question is more related to disease understanding. What do we need to do to the healthy cell to make it diseased? Obviously, we don't want to induce a disease in anybody, but if we get an answer to this question, it will at least be a good candidate to understand the possible root cause of a certain disease. Because believe it or not, we don't know the root causes of many of, in particular, chronic diseases. That's why they are chronic, because we are treating them like on the surface, but we are not addressing the root cause because the root cause is unknown. So these are a couple of scenarios in which we would do experiments in cells. So the general frame is we have a cell, we introduce some perturbation, and we see the effect of this perturbation. Or in a different framing, we take two kinds of the same cell, and we want to see what kind of perturbation takes this cell from one state to the other. And this answers the question, why do we do experiments in cells in the lab? So it still doesn't answer why do we need a virtual cell. And to answer this question, well, it's a kind of a short step. Doing this in the lab takes something on the order of a month. Some labs and some cases probably less, some cases probably more. So single experiment, single cell, single perturbation, and single outcome, let's say roughly 1 month of work. We have hundreds or even thousands, including different cell states, different cell types. We have for all intents and purposes, like infinite amount of perturbations and their combinations, because we have 20,000 protein coding genes, which we can knock out, knock down, try to amplify. We can add some known drugs, unknown drugs, and see how they work in different cell types. So to do this exhaustively in the lab, this is basically impossible. And this is exactly why designing new drugs takes so much time and money. So now imagine if you had a program that you could do this on a computer. You take a virtual cell, you introduce a perturbation that you want, you click enter or return, and you see the effect like almost instantly. Or you just run a huge experiment that perturbs all of the genes at once in a given cell, and maybe in all of the possible cells introduces some new perturbation, and just immediately observes the predicted effect and off-target effects. And this is exactly the idea we would like to have. This is exactly the idea of a virtual cell. We want a program that gets a cell state and a perturbation and predicts what would be the effect of this perturbation. In order to be able to answer the question, what happens to a cell when we perturb it, we need to somehow measure what happens to a cell, right? We need to be able to measure the cell's state. So how do we represent the cell's state in the context of a virtual cell? Well, like we would like to represent it completely. Okay, let's see an image of this is a diagram, a detailed image of a cell. Okay, this is a visualization of a cell. It's not an image, but it will do. Okay, this. I don't know if it's even an accurate visualization, doesn't matter, but it definitely reflects the complexity of what's going on inside. Okay, so we have different organelles, different ions, different proteins, lipids, just stuff floating around, opening, closing, rotating. And if we had this detailed representation, like a digital twin, perfect digital twin, then we could answer any question we would like. Spoiler alert, we can't. It's impossible to measure this complexity right now, or at least it's impossible to measure it at scale so that we get enough data that we can use to build a virtual cell. So what data can we use? Well, let's recap the central dogma of molecular biology, because this will be helpful here. So in any cell, you've probably heard that proteins are the things that do the work in the cell. Okay, and proteins get coded by the DNA, by genes in the DNA. So the DNA is the code that gets copied from cell to cell, and this is something that let's assume that it's unchanged. But then DNA can get transcribed into RNA in different amounts, and then mature messenger RNA gets transported out of the cell's nucleus, because this happens in the nucleus, and then in the cytosol, so inside the cell, outside of nucleus, ribosomes attach to the RNA and translate it into proteins. The proteins then fold, and not only the shape, but this whole structure, together with all of the forces, give it its function. And it so happens that we have a great way nowadays to measure RNA, the amount of RNA, and you have great ways to measure the amount of RNA on a large scale and on a single cell resolution. So we can measure how much RNA got transcribed of this gene in a given cell. This is called single-cell RNA sequencing, or RNA sequencing if it's not in single-cell resolution. And the amount of RNA is called gene expression. And this is exactly what we'll use. So this is now very useful to our, let's say, definition of a virtual cell, because we are going from a very broad, we have a cell, we perturb it, and we have a different cell state, to actually narrowing it down to we have gene expression, a cell represented by its gene expression. We introduce a perturbation, and we see how this perturbation affected this gene expression. So the amount of this gene that got transcribed in a given cell. So this will be our definition of a virtual cell going forward. One caveat to that is that it's actually technically impossible to measure those things, because the act of measuring destroys the cell. But we can take two copies of the same cell, two cells, let's say from the same colony, and we can introduce a perturbation to one and not perturb the other, measure one, measure the second one, and then observe how they are different. And this is exactly what we'll be doing. Okay, so we now know what we want to build. So now let's just build it. First, we'll start with an intuition. I have an open VS Code here. And this is like a completely empty directory called virtual cell. And we'll start by creating a virtual environment into which all of our packages will land. So we'll do it in Python 3.11, because all of the packages and deep learning packages and everything else is really stable here and works well also on my Mac. So let's just go with this. So we are creating a typical virtual environment. Okay, so now all of our packages will land in here. So let's start this environment. So it is active here. It's only active in the terminal right now. And actually we are not going to use terminal because I want to start building an intuition. So let's just build up the folder structure as we are going. So we'll start with source, src for source, and now node, books, and this is how I always start every project. Let's create a playground notebook, which will allow us to explore ideas. It already sees our environment, so it's already loaded up with the environment that we've created. So all of the packages will be loaded from this virtual environment. And now let's build an intuition for this virtual cell. So we want this to work something like we will have a cell with some identity, but let's leave it for now. And when we print the cell, it should print out its state, okay? And then we would like to be able to perturb it. We will say, yeah, cell perturb, and we would like to be able to observe that the perturbed state, how the perturbed state looks like. And yeah, this is actually a good suggestion. Let's say that we are going to simulate how a cell responds to hypoxia. Now, if we run this right now, except for the fact that we don't have a Jupyter kernel installed, so this will now install in the background. When we'll run it, it obviously won't work because, well, cell is not defined. We haven't created the virtual cell. This is the goal of all of this, right? But so let's build a very, very, very simplified version of this so that we know what we're aiming at and how we would do it if we had the knowledge and if we had the complete knowledge. So let's build a class cell. And the constructor for this cell is, yeah, but no. You remember exactly. Remember we said that we will represent the state of the cell and we will observe the cell, what happens in the cell, by analyzing the expression of genes. So you can imagine this will basically accept expression. And so you can imagine that this expression looks like we have gene 1, and it has some level of this gene, some amount of the mRNA of this gene, level gene 2, some amount of mRNA of this gene, and so on. So it will basically be a dictionary, just like it was suggested a second ago, but we want to be able to initialize it from the constructor. So here we would like to basically do exactly something like this. Compiler immediately recognizes that when you have hypoxia, those two genes will come into play, which is hypoxia inducible factor and PhD, which is encoded by EGLN2 gene. Because this hydroxylates this, so this basically attaches OH group to this, and this is then marked for degradation in the presence of oxygen. If you take out oxygen, this should go down, because there will be no oxygen to be used, and this will not be hydroxylate, so this will not be marked for degradation, and this will then go into cell's nucleus and work as a transcription factor for, well, hypoxia inducible genes. So we can simulate it here. So we'll do def perturb. We'll take some condition. Yeah, this is actually very good. This is exactly how we would want it to look like. And else, if condition is normoxia, no. Hyperoxia, we would do it exactly differently. Exactly the opposite way. Perfect. So now when we run it, okay, something's perfect. Now when we run it, this works and this works as well, but the output is like this, obviously, because we didn't print how the cell looks like. So let's just add how the cell should look like. No, let's just print the expression, and that's it. I think we can even print it like this. Or not. Yeah, okay. So now we can see that we created a cell. We initialized it. We said that this should be a cell with this expression profile. And it looks like this. Okay, now we perturb this cell. We introduced some sort of a perturbation, and we got, well, and now this cell has this transcription profile. When we now go to hyperoxia, we will go back. And if we did it twice, we would go back even further. And if we did it thrice, and so on. Obviously, this doesn't make sense. Only the direction makes sense. But it's a useful model right now. Okay, because it shows us exactly what we're after. Okay, we are looking for something that, given the input expression profile, will allow us to simulate, then given some perturbations, how this expression profile changes. Now, I wouldn't call it a virtual cell yet, because we have only 2 genes and 2 perturbations, which are basically 2 sides of the same coin. And in reality, there is actually 20,000 protein coding genes. This is only protein coding genes. There is lots of more transcripts available in expression data. And obviously the number of perturbations is even greater, because you can also, combine perturbations. You can do knockouts. You can do silencing. You can try to amplify some things. This is, and of course you can, pour some substances. You can put oxygen, iron. You can do a lot of different things. And doing it manually, like programming it manually like this, makes completely no sense, except for an illustrative purpose like here. So now we know what we are aiming for, because this is exactly what we would like to have, and this is exactly how we would like it to work. The only thing we need to change now is the inside of how it is implemented. And we're going to do this next. So now we've built up a case for what we want to achieve, because we want to have something like this, that we give it some initial expression profile, we give it some perturbation, and we observe the perturbed expression profile. But we definitely have built up the case as well that we wouldn't like to do it like this manually, right? And this is something where actually machine learning could be really useful if we had the data. So what data would we have, would we need? Well, exactly what we are using here. So we would need expression as an input, but this would be like an unperturbed initial expression. Then we would like to have perturbations, what perturbation was made, and what was the perturbed expression. Or maybe let's put it another way. We just want to have unperturbed expression and perturbed expression, but also for the perturbed expression, we would like to know what was the perturbation that was made here. And this will allow us to kind of create an equation of init exp plus perturbation equals pert exp. Okay, so this is exactly what our model will get as an input, and this is exactly what we would expect our model to get as an output, given as an output, exactly what we see here, right? So this is still a very, very useful model, but now we just pretend we haven't seen this, and we just need to implement the details of how this is done. So there is luckily a great resource for that in terms of data sets. There's actually a ton of data sets. There is one like by I think CellXgene, and it's not one, it's like a gigantic data set. But there is also Arc Institute Virtual Cell Atlas, and we are going to use that one because this is actually a collection of multiple data sets, which is really awesome. Here you can see there is a single cell base count, which is a collection of, when I last checked, it was 230 million cells. Yeah, 500 million cells. Okay, this is an awesome resource, but yeah, 500 million cells, that's a lot. But these are unperturbed cells, probably mostly. I don't know if all of them, but I would guess that most of those cells are unperturbed. Then we have Tahoe 100M, which stands for 100 million cells. So I'd say roughly it's, roughly 100 million cells. These are perturbed cells. I mean, controls and perturbations. So unperturbed and perturbed. But now this is actually a bit too much for us, because this is supposed to be like an introduction of how to build it, and this would require some more equipment trickery and technical things, which are beyond the point of the model itself. So we are going to use the Virtual Cell Challenge dataset. Virtual Cell Challenge was a challenge hosted by Arc Institute half a year ago, and they created a specific dataset for that of one cell type, which was human embryonic stem cell with 300 perturbations, so 301 because we had unperturbed and perturbed cells. And yeah, they give out this data for free, so we can just go in here. There is a tutorial on how to download it, and we can just do it. So let's just go with this. Okay, so first we need to install pandas, scanpy, and gcsfs. Yeah, I already had those installed here, but so now when we run it, this will be fine. And now we initialize the Google Cloud file system access. This is done. Now we have the address of the bucket in which the data resides, and we can build up the whole paths for the data. So this is the whole path for the training data, which actually the training data contains 150 perturbations, and validation data contains 50 perturbations. And now we can take this and we could just run this. I'm not going to run this out of respect for Arc Institute, because those datasets are not that small. They're not huge. They're not like terabyte. But there is probably something like, I don't know, 15, 20 gigabytes of data. And this is like a cost for them every download. So it's probably enough that you're going to do this. I already have this data on my computer, so I'm not going to just, run this to prove a point. So now I'm just going to move this data, because once you download it, you can save this. Save data to local file. Yeah. And I would actually do it. Like this. So obviously now if I'm going to run this, this is not going to run because I don't have the this variable, which contains the downloaded data from Google File System. But let's add data directory. And now I will now transfer those data sets here. And we can start from there. Okay, so now that we have the data, let's look at it. But first, let's clear out this notebook. And again, another convention that I always use, I would add another directory which would be called scripts. And inside of the scripts, I would put probably download data script, in which I would just put all of the code for, like, honestly, right now I would probably ask an LLM to do this for me. Just create a clean script, but let's just do it old school for a second, just to be quick. We don't need to present this right now. This is, we also want to do the same for val data path. And this is our data validation. And here we are going to save it. And validation data as well. Okay, so now we have a script that we can run whenever we want to. We need to download the data that we've used. And we can now just forget about it. Here we have some unused cells. Okay, so now once we've downloaded the data, we can now look at it. So let's open the data. Let's just do it like this. And we have, okay, well, I don't know, where did you take this from? Okay, so now we have the data, and it's an data object which has observations and variables, and it also has x. X is an array, but it's a sparse array. So now when we take the first row, it's, as you can see, the shape of this array is 221,000 by 18,000. So we have 18,080 genes, and this is the number of samples that we have. When we look at the first example, again, just a reminder, this is a sparse array. So what it basically means is that we don't keep zeros. And why is that? Well, because expression data is notorious for just having tons of zeros. So now when we densify it, we have the whole, this is just, as you can see, an array. This is only a single example, but if we take two examples, we will have a two-dimensional array. So this is the expression profile for the first cell, and this is the whole expression profile for the second cell. Now let's also look at the observation data, because this will be interesting for us as well, So now we have as many rows as we have cells. These are the guides for each of those, for identifiers for each of those cells. And here we can see what gene was perturbed in this cell. Here we have the ID of batch, if you wanted to use it, because this just means that those perturbations were done in batches, and batches have very often batches have something which is called batch variation, so some technical artifacts. So we could use this information so that we know that, okay, those samples come from one batch and those from the other batch, and we can try to do something about it. We are not going to do this in this tutorial, but it's something to keep in mind, that we have this information. We also have target genes, and this is the most important part. So let's look at target gene. And we can see that we have 151 theoretically target genes, but we also have non-targeting, which is the identifier of our control cells. So now, remember what we wanted to have. We wanted to have expression as an input. We also wanted to have perturbation. So look, here we have expression for, no, come on, man. We have expression for the first two, okay, I need to print this. Expression for the first two cells and perturbations for the first two cells. Okay, we said that we want to have unperturbed expression and then perturbed expression plus perturbation. So this is our output actually. So this is perturbed expression and the perturbation, and the unperturbed expression and our unperturbed expression, we will just read from this, right? So we know that that for instance, because the minus third target gene, the minus third target gene is non-targeting. Exactly. So when we now take data x, this is now actually, this is the expression profile of one unperturbed cell. So now we can see that basically we have everything that we need to train, right? Because we have unperturbed expression, perturbed expression, plus the information about what the perturbation was given to that given cell. Let's also look at one more thing, which is, because we said we have observations and variables, what do we have in the variable data? Here we just have the information about the 1880 genes that are in our data set. Here are the gene symbols, and this is their ensemble ID, so that we can cross-reference it easy. So now we know what data we are dealing with, how does the data structure look like, but now let's look also at the values. So let's take the first cell, and it will be data x0 to array, and also flatten, because this would be 2D and this will be 1D array. And the first cell looks like this now, right? We have 1880 expression values, but these are actually raw counts, because of how the single cell expression data is being collected. This is actually number of unique molecular identifiers, and different cells get sequenced to different depths, so different total UMI counts, purely just due to technical variation. So one cell might have 5,000 total UMI counts, and another can have 23,000. But this difference tells actually nothing. It's completely not related to biology. It's just a sampling artifact. So we need to correct somehow for this, because otherwise the cell with more counts would appear to express every gene way higher than the cell with lower total count. So there is a simple thing we can do. We can basically we can basically normalize to the common total number, and this is often called CPM, counts per million normalization, even though it's not always normalized to million. So how does it look like? So we take this expression profile that we have, we divide it by the sum of all of the values. So now we normalize it by the sum, and we express it as and we rescale it to a different but a common total across all samples. So now, and this is called and there is a function for that in ScanPy, which is called SC preprocessing normalized total. And this is exactly the same. So let me illustrate that it's exactly the same. So let's do a data copy. And now we have normalized total. And we have the first cell now will look like this. Okay, as you can see, I didn't change anything, and it looks exactly like that. But let's just do it on the copy as well. So here is the first cell, first copy cell, exactly. And let's just do first copy cell. First copy cell will look like this. And now let's just compare those two. So first cell equals first copy cell. Yeah. If we do a difference, we don't have NumPy, but maybe we have just max. Yeah, so the maximum discrepancy between those two is on the order of 10 to the negative, actually almost 10 to the negative 8th. So at 8th place after the comma, which theoretically could be a lot, but since our values are actually in the range of, the min is 0, congratulations, but the maximum is, so the order of our values is from 0 to 100. Well, this basically tells us that we are here at the floating point precision, because floating point precision actually represents like 7-8 digits. Float 32 represents 7-8 digits precisely. So this would be correct. So now you can see that this is basically exactly equivalent to doing this. So we're just going to do this. There is another point to be made here, because let's just remove this, let's clean this up. And now we have the data that's already normalized. So let's plot data. Plot the distribution of the first row of the data. Let's use matplotlib. Okay, let's just do it like this. Okay, this is not what I had in mind, because we actually want to skip zeros. So the first cell would be and then, but here we want to just do first cell, but only the values that are greater than 0. Okay, so now we can see that even though the values are on a slightly probably different scale that there were, the distribution is extremely skewed, and the range of values is also very high. So what we also do to also kind of decrease the range, but it's not the only point, but to decrease the range, we take a logarithm of this data, because we can see that it's extremely skewed. So when we take logarithm of this data, and this 1p, it just means that it's actually natural logarithm of the data plus 1. Okay, so now when we take the logarithm of this data and we plot it, the distribution looks way nicer. The range is way more friendly, and also as an important effect is that the variance is stabilized across different genes if we do this in the whole data set. And this will really help visualizations, but also training, in training the model. So those two things, those two operations, just normalization to a common total target sum, and taking a logarithm is something that we'll be doing to our data before actually using it for training. Okay, so now we have everything we need in its right place to start training. So let's create a script. It will be train py, and let's just go through what we need to do. First, we need to load data. After we load the data, we need to normalize the data and log transform the data. After we do that, well, we can create a model. Actually, we will here create an optimizer, define the loss. We'll go to it later. And then we'll have loop through epochs. And then we have train, loop, and validation loop. Theoretically, at the end, let's actually indent it as it was suggested, because at the end we should also do a test loop, but we won't do this because the point of this, the first step is just to unclog the pipe, just push through the pipe till the very end with the simplest steps possible, minimal code, very easily understandable, but still as little as possible code, as little dependence on like external functions, features, stuff like that. After we've unclogged the pipe and we actually have a working something, but something that actually works, then we can start making it nice, adding features, expanding the model, separating out parameters, separating other functions, taking datasets and making them, again, prettier. But first, let's just unclog the pipe till the very end. So we need to load the data. First, we import this, and this will be data equals sc read. Perfect, right? This is our training data. Now we normalize and log. So sc normalize total, as we've talked about, and log. This is all we already know this, what happens here exactly. But now before we create the model, actually, we also need to split the data into training and validation sets. As you can see, we've downloaded both training and validation data from the competition. However, we won't have, we are not participating in the competition, so we will treat the validation actually as a test data. And we will split our training data into training and validation so that we tune the hyperparameters and choose all of the parameters and hyperparameters of the model with only on this set, and then we will test how it would behave in the wild on the validation data. So we need to do a split. Split data into train and val. And let's say that we want to have split, we will have like 80% of samples in the training. So, well, normally we would split the data just across samples. We would take randomly selected 80% of the samples and take it into training and into validation. Yeah, exactly what is suggested here. This is wrong, right? And why is that? Well, because we actually have not only samples, but also, or primarily, we have perturbations. So we actually have genes A, B, C, D, and E, let's say. Let's say that we have only 5 perturbations. If we split the data across samples, and so we have like, let's say we have 100 of samples, but 5 perturbations. If we split the data across those samples, there is a very, very high probability that the training data would see exactly the same perturbations as the validation data, just by random chance. Or more precisely, we have absolutely no control over it. It's just, probably it would be like that. But in our training data, we've seen that we actually have, let's just load it, and our data, target gene, not value counts, we actually have only 150 perturbations in the training data. But we also know that, well, data X, we actually have 18,000 actual genes that we would like to predict perturbations for. So this is a big discrepancy, and our model needs to be prepared, and we want to evaluate it on a task of predicting perturbations to actually unseen data. So what we would like to have here is something like, we will train the model on perturbations A, C, and E, but validate on only perturbations B and D. So we cannot split the data across samples. We split the data across perturbations, genes. So let's do it, okay? So first, let's just take genes. let's take all of the genes. Genes is data observations, target gene, unique, but we also want to exclude from that the non-targeting gene. So we can do it like, we can work on sets, and then we can exactly. Okay, so now what this gives us is, let's look at it. Yeah, great. This gives us exactly 150 perturbations, excluding the non-perturbed genes, because the samples, because we don't want to predict them. So now we have all of the genes that are actually perturbed. So now we can split them into training and validation genes. And let's just do it randomly. Let's random seed will be 23. NP, let's import NumPy. And let's just do NP random seed, perfect. And now we want to select training genes, and exactly. Okay, so training genes is a random choice, random sample from the list of all of genes. We want to sample 80%, right? This is 80% of all of the genes without replacement, right? We don't want multiple versions of the same, we don't want the same gene multiple times. We want to pick every time different gene. And validation genes are just all of the genes except for the ones that were used for training. Perfect. We have the gene split. Well, but that's not it. This is only gene split. Now we actually need sample split. But the sample split needs to be done according to the genes. So let's first do also we need control indices. So this is control indices. Train indices is train genes. I don't know why this. No, this is not right. And validation indices is, so, okay, what is this right now? Let's make sure that we understand what happens here. What's happening here? Let's hard code it to 0.8. Okay, so we have now true on samples of all of our 220,000 samples. We have a vector now that has false where there is no control index, and there is true where there is a control sample. And the same is true for all the remaining data sets. So we have training indexes here, and we have validation indices here. And when we like sum all of those, those all will be true. So these are all mutually exclusive, and they all sum to 1, sum to the whole data set. So now that we have indices, well, it's very easy to pick the data sets. So we have X control is just data X to array. Why to array? Because it's sparse, and for training we actually don't want to. In this, again, we are making it very, very simple without any optimizations. So that's why I'm just bringing it straight to dense format, because like by default, it's in a sparse format. If we did it like this, that X control is sparse. If we do to array, it's immediately in the format that's like digestible by the by PyTorch. Okay, so we have control samples. We also want to have obviously train, expression, and X validation expression. Yeah, but that's not it. That's not all. We also need to remember, as an input, we have control expression and perturbation, and we want to actually predict this. So maybe this is a wrong way of putting it. This is actually y, and x will be x control expression. You know what? No, let's leave it as it was, not to confuse ourselves too much. We will just call it perturbation train, and this is exactly perturbation train, and then same perturbation validation perturbations. So now perturbation train is a list of all of the training samples, of all of the training perturbations. But remember that when we take unique perturbations, there will be only 120 of them, because we did an 80% split. So there are 120 unique perturbations, but they are across all of the 150,000 training samples, because training indices, when you when we sum the training indices, this is exactly the number of training samples that we have in our dataset. Okay, perfect. So now we have split the data into training and validation. Training and validation. And for each training sample, we have control expression, which control expression is separate, and we will be sampling from control expression samples, from non-perturbed samples. So control samples just completely randomly. And from those control samples, plus a given perturbation, we will try to recreate the perturbed expression. Let me call this y, because this will be more appropriate actually. Even though it's called x, but this actually y. This is something that we are trying to predict. So now, well, it seems that we have almost everything. We have everything. But there is one catch. And the catch is the model needs to accept numbers. We are going to use, I already spoiled it, we are going to use PyTorch. So we're going to use like some very, very simple, in this case, neural network, a multi-layer perceptron. But a multi-layer perceptron needs to accept numbers. So as you've seen here, well, we don't have numbers, right? Training samples, our training data, are expressions, right? These are this is expression data, so these are numbers. But also at the same time, the perturbations are strings. These are just like random strings of text, of characters, at least from the computer's perspective, right? So where's the problem here? We need to somehow convert those strings, so those symbols, let's say, into numbers. And the easiest way to do it is like this. It's called one-hot encoding, and you've probably heard of it. So let's say that we have, no, thank you, no. Let's say that we have 4 genes, A, B, C, D. So the easiest way to encode them is to say, since we have a dictionary of only 4 genes, we can say that the first gene, gene A, will be encoded by 0, 1, 0, 0, 0. The second gene will be encoded by 0, 1, 0, 0, and so on. Why? Well, because this just corresponds to A, B, C, D. Okay? So now this vector, whenever we use this vector, it will represent gene A. Whenever we use this vector, it will represent gene B. So now theoretically, the input to the network is actually expression plus this 1000 perturbations. Okay? This would be the input control expression, and this is also numbers, right? So, and a concatenation of those two, combination of those two numbers would be the input to our model, and we expect it to output some expression. However, there is a catch, because this only works if the model will see the genes that is later being evaluated on. And we've established here at the beginning that we don't want that, right? We want the model to train on one set of genes, but then be evaluated on another set of genes. Because in our case, we actually have 120 training genes, and then we have 30 validation genes. And then we actually have 30 validation genes, but actually we have a total of 18,000 protein coding genes that we would like the model to work on. So if we only do this and do one-hot encoding for the genes that we have in the training set, this would only go up to, well, 120. And then we show the model another gene, and it will do, like, it won't work, right? Because it won't have a correct mapping. And even if we expanded this mapping into 18,000, so this goes really, really, really long, and this is 18,000th zero in this case. So we would have, for A, we would have 1 and almost 18,000 zeros. And we would show those vectors in the training. In the validation test and just in general real-world performance later, it would, if we gave it some gene that was completely unseen in the training data, it would have absolutely zero chance of working in it, because it would never have seen 1 in the place corresponding to that gene. It would only see once in places of those 120 genes that we actually had in the training data. So long story short, we cannot do it like this. Okay, so what else can we do? Well, we can use embeddings, and we're just going to talk about them next. Okay, so what are embeddings and how we're going to work with them? So given what we just said, let's say that we have a dictionary of A, B, C, and D genes. We want to represent them as a fixed vector of numbers. Let's say that they will be of length 3. And those will represent something or some things about those genes in such a way that it also reflects relationships, or primarily in such a way that it reflects relationships between those genes. So let's actually go through embeddings using an example. Let's look so technically now you know that an embedding will just be a vector representation of our gene. This will be a gene embedding in particular, but we have embeddings. You've probably heard of embeddings, in particular in the context of text and LLMs, because all of the text models and all of LLMs use some sorts of embeddings for the tokens. So each LLM probably has a dictionary of something on the order of, I don't know, 60,000 tokens. I'm now guessing, but let's say that it's on the order of 60,000 tokens, which is actually something similar to genes, because we have something like 60,000 transcripts that can be read through single-cell RNA sequencing. So it's very, very similar order. But then those tokens are represented in way, way, way lower dimensional, fixed dimensional space that preserves relationships between the tokens. And let's see embeddings. There's a nice illustration of embeddings by Google on Google's website. Yes, great. Okay, this is perfect. So it's an illustration of embeddings on food. We have some foods here, and here is an illustration of one-hot encoding, exactly, right? So for instance, borscht, we can encode like 1 and then all of the zeros. Hot dog will be 0, 1, and then all zeros, and so on. Exactly the same as I just described for genes. But now, how embeddings work is that they put all of those, in this case foods, in our case genes, on some axes that represent something, represent some properties of those items. In this case, let's say, for instance, we have foods, and let's say that we have only one axis that represents the sandwichness of a given food. Okay, so we go from less sandwichy foods to more sandwichy foods. But obviously, in terms of foods, and as well in terms of genes, we can have way more dimensions representing some characteristics of those items. So for instance, we can add, here we have the sandwichiness, but we can also have dessertness. And then Apfelstrudel would have very, very high dessertness compared to all of those, but it may be kind of high also in sandwichness. Here we have another dimension. Okay, so we had the sandwichness and dessertness, but we also added liquidness. And now the soup obviously wins in this dimension, but we see now the relationships are becoming more and more complex. And what's very, very, very important here, as perfectly illustrated by this example, now we add some other food that we haven't seen before, and by placing it in this multidimensional space, we, even though we haven't seen it even in training data, we can still kind of see what's its relationship to all other foods through this space. So this is a perfect illustration of what are embeddings in general, and this is exactly how you can feel, how you should think of them, but in terms of genes. So in terms of genes, you can think of them, okay, do they participate in the same processes? Are they on the same biological pathway? How involved are they in metabolism? How involved are they in respiratory metabolism? How involved are they in inflammatory signaling? So all of those different dimensions are encapsulated in the embeddings. And here we only see 3 dimensions, but we will be representing our genes in, of course, way more dimensional space, but the principle and the thinking behind it is exactly the same. And for gene embeddings in particular, there is a great resource, gene embeddings. There is a paper that was out last year. Okay, I don't think that this matters if we take the current or the previous version, because we actually are not after the paper, even though it's amazing, like highly recommended to look through it, because it benchmarks existing gene embeddings on different prediction tasks. And actually, I think only something like 38 different gene embeddings. And what's even better is that they provide all of those embeddings for download. So all of the 38 embeddings that were tested here are very easily downloadable from just this single place. So you can just hit download. I already downloaded it, so I won't be downloading it twice. So you can download it, unzip it, and then we will choose which embedding we are going to use in our particular case. So as you can see, after unzipping it, you can probably see that there are many different embeddings. We are actually going to go with ESM2. Even though in the paper, GPT Model 3 embeddings scored highest the most, they are based on text, and so they will be probably on either. So they are based on what is currently available, and they will represent the genes to an extent that is currently well described in the literature right now. So more obscure genes will probably get very, very, very little description. That's why their representation will be very poor. The ESM2 is based on a protein language model, and it's a model that uses actually embeddings for amino acids. So it's a pooled mean of sequence of amino acid embeddings. So this more reflects the actual structure of the protein that will be created from a given gene. So we're going to go with this because this is just cleaner, I think. Here we have two files. We have genelist.txt file, and those numbers here represent gene IDs from NCBI. And this is basically a matrix. Okay, I won't open this here because it's 300 megabytes, but this represents the embedding vectors for each of those genes. So each row here corresponds to one row here. So the first gene is, let's see it, NCBI. Yeah, as you can see, this is the ID of the first gene. This is some gene. And from this, we can then infer what's the gene symbol. And then we can match those with our perturbations and treat this vector, those vectors that are here, as inputs to our model for a given gene name. Okay, now we have everything in place, so we can just build the whole thing till the end. Let's just remove this. Let's start by releasing memory from this main table because we don't need it anymore because we've split it into training and validation sets and control samples. So this huge array is not necessary here. By the way, like normally we wouldn't do it like this because we wouldn't be able, with the amount of single cell RNA sequencing data available, we wouldn't load everything into memory anyway. We would do it in kind of batches. So we would take up a batch, reload a batch, use it for training, for validation, then release it, then load another batch, and so on. But here, since we want to keep it as simple as possible, we are loading everything at once into memory. And that's why after splitting we can just release the main table. Okay, so now let's build it. Let's actually go from top to bottom. So we will just complete the whole thing, but with high-level code that will not work because we will need to then implement the details. But the high-level workflow should already tell us how the details should be implemented in exactly. So the details will be basically auto-completable. Let's try this. So first we have the data and we create the model. Okay, so the model will be a virtual cell model. And yeah, what do we want to give it as input? The input would be, remember, expression dimension. This will be both an input and an output, okay? So this will be the shape of the control expression. Yeah, exactly. Then we will have embedding dimension, which will be, well, exactly as the embedding dimensionality is here, but we don't want to like hard code it from this file, even if though we could. We want to make it general, so once we read the embeddings, we will fill this up with the dimension of the gene embeddings, but the second will be embedding dimension. Mind you, the expression dimension is both the input for the control and also what we are aiming to predict. So this is also, the input will be expression dimension. So the architecture, if you will, of this simple net will be something like expression dimension plus embedding dimension. This will go into exactly one hidden layer to keep it as simple as possible. And then we will have as an output expression dimension, this is the target, right? This is control, and this is the, so this will be from here, and this is from here or from here. And embedding dimension will be based on the encoded perturbations. So now what we are missing here is, as you can see, we have expression dimension, we have embedding dimension. The only thing missing is hidden dimension, which will be something as well. Hidden dimension, exactly. That's it. This is our model. Actually, we can make the hidden dimension like a hyperparameter, but that we control from here. Let's make it 512. And actually, we can do it like this. Yeah, okay. So we have the hidden dimension, the expression dimension, and the embedding dimension, which we will infer from the embeddings that we'll read later. So this is our model, perfect. Now we need to choose an optimizer. We will just use Adam. We're not going to use weight decay for now, so it doesn't matter if it's Adam or Adam that treats weight decay differently. Let's import torch. Perfect. So we have optimizer. Now we want to calculate the define loss, to define the loss. And now we could use mean squared error, but let's go even simpler. Let's just do L1 loss. And one loss is basically just mean absolute error. So let's also set manual seed here, and let's also import torch and perfect. Now we have the loss defined. So just to make sure that we're on the same page, if we have like 0, let's say predicted will be 0, 1. That's not 1, but never mind. And true would be something like this. The loss is, well, pred minus true mean. That would be the loss. So this is 0, 0 means 0 minus 0 is 0, 2 minus 1 is 1, here is 0, here is 1, and here is 1 as well. So we have 3 divided by 5, so it's 3 divided by 5. So 60% loss, 0.6 loss. So this is our loss function that we will use to evaluate each prediction. And now we basically can just go into training. So the epoch loop, exactly number of epochs. let's call it max epochs for now, and let's set it to 10. And let's just iterate over that. So for each epoch in max epochs, okay, we're going into training loop. Model train. And here, yeah, of course. Now we want to iterate, yeah, but not like that. I told you we want to go high level. So we would like to have something like we want to iterate over batches of batch control expression, batch perturbation, and batch target expression. Exactly, in the training loader. So we want to have something like a training loader, a class, we'll go there, wait a second, a training loader that will basically give us, based on the batch size that we will define in a second, batches of control expression, like randomly selected control expression vectors, and perturbation embeddings and target expression that are paired. Okay, so one perturbation here corresponds to one target expression here, and it should be derived from single control expression. Here we zero the gradients from the previous iteration, and then the model, as we defined, we want to give the model the control expression and the perturbation. And the model should predict the expression of this perturbation, after this perturbation, in a given control cell. Then we calculate the loss, and then we do the backpropagation. Yeah, we also need to record the loss. So that we'll have training loss would be, yeah. And now training loss, exactly. Why do we do this? And afterwards we do something like, yep, exactly. Why do we do it like this? Well, because we are not guaranteed that we will, patch, perfect. If we divide the dataset into batches, let's say that the batch size will be like something like that. Let's not confuse ourselves. And let's do it different than hidden dimension. So this is our batch size. It doesn't matter, but just not to confuse ourselves. This is our batch size. So it will be very rarely the case that the batches perfectly split into exact batches of 256. The last one will most definitely be shorter. So that in order to calculate the average training loss properly, we sum the weighted losses of each batch, but then divide by and the weight is basically the size of a given batch, which will be the same for all of the batches except for the last one. And this will ensure that our training loss, that our average training loss is calculated correctly. Sorry. Now we can go into validation. Validation loss is 0, model eval with torch no grad, and we do exactly the same. And also validation loss. Perfect. So since, as you can see, this is also like a meta learning. Once you define all of the abstractions correctly, so once you express what you actually want the code to do, the autocomplete part is working way, way better because it gets more and more constrained on what is expected of this code. And now we just want to print. Yeah, exactly. We just want to print what epoch are we at for each loop. For each epoch loop, we want to print what epoch we're at, what's the training loss and what's the validation loss. And also, not even finally, we want to save the model. I wouldn't do it like this. Let's do it by runs, not import run, but we'll do something like run equals date time now. Yeah. Okay. Now, why do we do it like this? Because now every time we run it, we will save the model dictionary, the weights of our model. But if we run the model twice, we will not overwrite our previous run. This is something that comes with experience. And I just want to make sure that I never, when I run the code twice, I never overwrite my model, because obviously I've been there, that I ran the model that was like trained for a day or two, and I ran training that overwrote the previous model just by accident. So this just makes sure that every model is different. If we want to continue training, then we would tweak it, but then I have to be very deliberate about it, and now it's almost like it's impossible to do it by accident. But we also don't want to write only the state, the weights. I would like to write the model config. So model config, I would write it something like this. Exactly. So let's change this now to exactly. And now we can model checkpoint both config and weights. I prefer the other order, even though it doesn't matter. Yeah. So we are writing both how the model looked like. So probably, even though the expression dimensions and the embedding dimension won't change, the hidden dimension and in general the hidden parameters may change, definitely. So we're just making sure that we can reproduce it all, even if we later change the parameters here. Okay, so now almost everything is in place, except for, the model. So let's just write it. The model will be a class, but as I told you, this should be straightforward right now. So let's create a class virtual cell model. Yeah, we'll have to fix it, but let's work with that. So we have expression dimension, embedding dimension, and hidden dimension as inputs, as we defined here. Expression dimension, embedding dimension, and hidden dimension. They all go into the model constructor. Now, the first layer gets the embedding dimension and the expression. It completely doesn't matter, but I like to keep the order the same. I don't know why it was flipped here. So this is the input, both vectors together. They go into the first layer, which outputs are of dimensionality hidden dim. Then this goes into the final layer, which is the output layer, and the hidden dimension gets blown up back to expression dimension. So now in the forward pass. So when we call the model, we give it control expression and the perturbation embedding. Control expression and perturbation embedding. We actually give it perturbation embedding here already. So now we concatenate those two, which basically means we just glue those together. So we get one big vector. And this gets fed into the first layer, which then goes through the activation function, which is ReLU here, which is perfectly reasonable. And this will be of dimensionality, hidden dimension. Then this goes into the second and final layer in our very complicated network. So this is hidden dimension, of dimensionality, hidden dimension. And it gives us out the expression dimension, which is the predicted expression. Great. So now we have the model defined. We need nothing more from it right now. This model will learn something. So now what we need embedding dimensionality, but we need training loader and validation loaders. Yeah, so let's just build them. So let's just define them here. This would be appropriate. So we need train dataset first, and it will be perturbation datasets. And what would we like to give to this perturbation dataset? Control X, definitely. Perturbed perturbation symbols, so genes that we want to perturb, and the expression that we expect at the output. But we also, remember, those are symbols. We downloaded the embeddings so that we can translate them into vectors. So we also need something like gene to embedding something, okay? Something that will take those perturbations and return embeddings for each one of those. Gene symbols and return embeddings for each one of those. So this is perturbation dataset. Training dataset, validation dataset looks exactly the same, but we just have validation perturbations and validation predictions. And now we also need a train loader, which will do exactly this for us. Okay, so the train loader will call the train dataset. Here we are using the batch size, so this is also handled by us, and we are shuffling the examples because we don't want the order to impact how the model is trained, because we'll probably train it for like very large number of epochs, and we don't want the model to see all of the examples always in the same order, because it may impact how it learns. But in terms of validation, it doesn't matter. But even the other way around, we want the validation to be as consistent as possible, so we don't want to shuffle the examples when we're validating it. Okay, data loader is easy because data loader is actually from Torch. Exactly. But we also want data set here because we are going to define data set here. And the data set will be a class perturbation data set. Okay, let's see if it knows what it's doing. So our perturbation data set is initialized exactly as we defined, okay? So we define it with control expression, symbols to perturb, and the expression, the target expression, and also gene to embedding mapping. We don't have it yet, but let's just see if we had it, if this was a perfect dictionary that gets symbol and outputs like some vector, this would be great. Not some vector, but the embedding vector of a given gene, this would work. So we'll work on that in a second. But first, let's work if it works. Let's see if it works if we had it. So we are just storing the control expression immediately as a tensor. Great, so that we don't have to like convert it every time we ask for it. Perturbations, genes are just stored as they are. Target expression also perfect, and gene embeddings, gene to embeddings also great. This is just for Python to be able to determine how long is the data set. So we are overwriting the length, and this will be called for every time it tries to sample. It will give some index of the length of this whole data set, and it will pick samples from here. So this basically gets called whenever PyTorch wants to sample a single sample for training. And let's see if it's good. Okay, it's not. Because we immediately see that the control expression is picked by the index, and it shouldn't be. So let's just go through it step by step. First of all, let's retrieve the perturbation. This is correct. Okay, so we select one sample. This will be some random number between 0 and the length of number of perturbations -1. And we sample a given gene that is being perturbed here. And for that gene, we want to extract the embedding. So we use our dictionary to extract the embedding. We convert it to tensor, torch tensor, and we have it here. Perfect. Now target expression is already a tensor, so we just look it up by the index, by the same index, because this target expression corresponds to this gene perturbation. So this is great. What's missing here is the control expression, because we actually want to randomly pick control samples from which we will predict the target. Remember, this is just a single cell type. We only have control samples. Okay, so theoretically, we could even do something like average all of those and always pick the same one. Okay, this could be one, because this is basically one unperturbed cell. It just so happens that those two cells are identical. That's why, in order to show the model some like variety, we are picking randomly control expression from the pool of control samples that we have. But we want to do it randomly. Later, we will actually do something like batching or mini batching them to make the training more stable. But again, here, we just want to make it as simple as possible. So, and now, yeah, the perturbation dataset is done. We are returning the control expression that we sampled. We are returning the embedding of the perturbation we are aiming to predict, and the target that we are trying to achieve. So this now mimics this perfectly, right? We have training loader, we have batch control expression, batch perturbation, let's even call it embedding to be more verbose and precise. And here we also want to change this to embedding. So now, again, we have batch control expression, batch perturbation embedding, and target expression that we are trying to predict. And we feed the model with control expression and perturbation embedding. And then we evaluate the predicted expression on the target expression that we are trying to predict. What are you talking about here? Yeah, this is something that we need to fill up as well. And exactly, yeah. So what is this? This is the dimension of the embedding. This is the only missing piece right now in this whole code, is the gene to embedding dictionary, and yeah, let's just do this next. Okay, to do this final piece, let's actually move to the playground. Okay, we have two files. We have the gene list with NCBI gene IDs, and we have the corresponding gene embeddings. Okay, so each entry here corresponds, so this vector this is the first line. Okay, great. The first line corresponds to the first ID, or from the other way around, this ID corresponds to this embedding, the first line. So now we want to obviously read this, so let's just do it. First, we'll read the embeddings. We'll use pandas for that. Embeddings will be just pd read. Yeah, close. As you have seen, there is no like headers, indexes, nothing here. So let's just do header none. And also, let's copy the path. Just copy relative path will be easier like that. However, if we are in the notebook, so notebook, in the playground notebook, which is inside notebooks, which is inside source. So that's why we need to get out of here. And our embedding looks like this, okay? So we have embeddings for 18,902 genes, and we have the dimension of each embedding is 1,280. So theoretically, we could just hard code it here. But, well, if we decide to change the embedding later, this is generally it's better not to hard code stuff that you don't need to hard code. So I would just leave it as it is. So once we have the dictionary, we will just read it from the first row, from the first embedding. So we've finished reading the embeddings. Now we want to read the IDs as well. So the IDs will be with exactly, but let's copy the relative path again. And yeah, let's say these are IDs, not genes, okay? So with the IDs now, IDs are exactly that. And the final thing we want here is have a dictionary that would give us ID to embedding. Okay, so for each ID, we are returning both the gene ID and its corresponding embeddings. So now ID to embedding is something like this. Okay, so now when we take some gene by its ID, we get its corresponding gene embedding. Perfect. Close. I don't know if you remember, but we don't actually have NCBI gene IDs. We actually have gene symbols. So the final thing we need to do here is to we actually want to have symbol or just gene to embedding, not ID to embedding. So this is our goal. So that here, if we wrote ACSL4, it should give us the embedding of this gene. So now there is a library called mygene. And this is how you initialize it. And using this library, we can query the database, an online database that allows you to translate between gene symbols and any kind of gene ID. You can use the CBI ID, you can use ensemble ID, any kind of ID for any species. You can then cross-reference the genes and translate, like in our case, IDs to symbols or symbols to IDs. So this is exactly what we need to do right now. So we are just going to query many, and we are going to query the no, really not like that, man. We are going to query interest gene. This is the NCBI ID, otherwise known as interest ID. We want to retrieve for each NCBI ID gene symbol, and we want to do the mapping for the human species, because the mappings actually exist for many different species. We want it to work for, we want to do it for humans. So now we run it. It runs a query to the database, and it will take a second. So let's just wait for that. Yeah, not that bad. It was just 38 seconds. Now let's look at the result. And the result is a dictionary of length, not a dictionary, but a list of length, 18,903, which is coincidence. I don't think so, right? It's just the exact amount of number of IDs that we queried for. So for each ID, we got a response. Now, how do we get from this? We want to go to gene to embedding. So now let's just go from gene symbol to gene ID, because remember here we actually have gene symbols, right? Going into the data set. We have perturbation genes, so these are gene symbols. And based on gene symbol, we want to retrieve the embedding. So we have ID to embedding. Now we want to have a gene symbol to ID, so that we want to have, so we can use ID to embedding. So we want to have gene to ID, and it should be something like this. This is actually not bad. Okay, so for each item, if symbol in item, okay, let's not overcomplicate this unnecessarily. So for each entry here in this result list, for each entry in this result, we want to have the symbol as the key. The symbol is the gene symbol, and the ID, which was our query, as the value. This actually makes sense. Let's see how it looks like. Yeah, exactly. So now when we type in, we get the precise ID of a given gene symbol. So now the last thing we need to do is to go to gene to embedding. And let's just do this methodically, okay? We want to have a gene as the key, and the value is the ID to embedding of gene to ID of this gene for every gene in gene to ID. Yeah, that was unnecessarily long. Now, when we look at this, we can see that we have the same, still the same length of the dictionary. And yeah, this should just work, because now for gene to embedding, does it work as it was supposed to work? Exactly. So we input the gene symbol, and as an output, we get the embedding of a given gene. All of the embeddings are exactly of the same dimension as they should be. So yeah, now we can just make this look nice. So let's tidy it up. So let's just put it into a single function. Get gene embeddings. Let's just work with path, and that's it. And hopefully, given the code above, it should be just copied, but let's double check like to be sure. We are reading the embedding using the path. Actually, okay, never mind. OS path. I would actually prefer it like this because this is cleaner. I know we have to like import OS, but we probably did it anyway in our code somewhere, right? Not yet. Oh, never mind. So, okay, we load the embeddings. We load the gene IDs from those two files. Then we create the dictionary that turns gene ID into embedding, so fetches embeddings given gene IDs. Then we query the mygene database. We give it NCBI gene IDs and take symbols. We do the gene symbol to NCBI ID mapping. And then using those two mappings, we can have a single gene to embedding mapping, which we now also can save because we don't want to like bombard the that server unnecessarily. We can just cache it. So let's also do check if cached embeddings exist. Yeah, we can do it like this. So let's do it like this. So first, when we run it, we check if we already saved it before. Okay, so this is our cache path. Actually, this is, I don't want, maybe, okay, let's leave it at that. Let's leave it like that. And if it exists, we just load it from the file and just don't do this. But if it didn't exist, this will execute. And now we will cache the embeddings. So we just want to save the gene embeddings into cache, basically just drop the embeddings to a file. Let's see if it works. So when we do gene embeddings, so get gene embeddings, this is our path. This should again take 30 seconds. So in the meantime, we can basically copy it there. Now we need to add a bit more stuff, imports. Let's just tidy it up a bit. Okay, this should be done right now. It is. So we have the embeddings. Yeah, this is gene to embedding actually, but we called it like this. And now we can query. Perfect. Works. So now we already copied it there, so we don't need those things here. Let's go back to training. And now we just need to use this. Because we need it here. This is the place where we need it. But actually we can load it earlier. So once we loaded the data, we can load gene embeddings. Exactly. Perfect. Beautiful. So we have gene embeddings already here. And now this should work. Okay, one more point is that the genes, we need to make sure that all of the genes, because remember, here we have some perturbations. We have actually 150 perturbations, but this list is completely like independent from the list of perturbations that we have here. So it may happen that we will not have an embedding for some perturbation that we actually need. So let's just make sure that we will basically skip those in training. I obviously already checked it before and we only have one case like that, but just to be sure, it would be a good practice to check it first. And here we just need to make sure that we only train on the perturbations that we have embeddings for. So here we have the genes. And here we need to filter the genes with embeddings. Exactly. And this is one way of doing it. So we are only selecting the genes if the gene is present in the gene embeddings. That's it. Now we can actually print it out, so spell it out so we know what happens when we run this, because this is now ready to run. So let's print loading data, loading embeddings. Yeah, no, loading embeddings, thank you. So this is print, this is not splitting data. Total perturbed genes. Let's write it here. Total genes, okay. Here, genes with embeddings, okay, so that we know exactly how many genes are in each how many genes we had in total perturbations, 4 perturbations, and how many genes like survived the embeddings filter. Let's print also the train genes, the validation genes. How many are there? There should be 120 and 30, but we know we'll probably skip some due to like lack of embeddings. And now what can we print more? We are printing every epoch. We can also be verbose here and just display progress bar for each embedding, for each epoch, at least for training, so that we have a ballpark of how long it's going to take. So also we want to import TQDM. And yeah, I guess we are now ready to run this. Fingers crossed. Loading data, great. Yeah, it takes some time. So yeah, exactly. We have a total of 150 genes. Genes with embeddings, we have 149. So we don't have an embedding for one gene. We have then 119 genes in training, 30 genes in validation. And this is the training, just flying. It's not actually flying, but it's okay. I think we could bump up the batch size. And okay, we have some training loss and some validation loss. Let's see if that actually goes down. If it does, we have something. Well, we have something, okay? We have a model that learns to predict the expression with some accuracy. And we see that the model actually improves with each batch, and that both training loss drops and the validation loss drops. Probably in this iteration, the training will surpass validation, not yet. So still validation loss, training loss is still higher than the validation loss, which is good, but it will probably change in a second. And we want to make tons of tweaks to this. But yeah, we have a working training loop for a virtual cell. How cool is that? After the training is complete, we have our model. We have our model here, and now I just realize we didn't remove this epoch here. So let's just remove it because it doesn't make any sense here. This is just model with this, let's say, model ID. Let's just also move it to, let's say, models. Let's create a directory models. We could call it output, or we could call it whatever. Doesn't matter. Now just all models will be placed here. So everything is nice and clean, or is cleaner. And about clean, we've basically now just unclogged the pipe, pushed through the whole process, start to end, at least for creating. Let's now also make this script a bit nicer, and general this code base. So we'll do it by just moving things to different directories and turning this into a package. So the train will probably stay here, but we want to move those into their corresponding files. So let's add another directory that we'll call like ML, and we'll in ML we'll have init.py. Here also we'll have init.py, and here we will have something like utils.py, and in utils we will put get gene embeddings. So we have get gene embeddings here. We add the imports, import OS, import my gene, and import pandas. Perfect. Now, so this is now gone. We want to move this as well to a separate file, and actually can be here in ML. It makes sense. So let's call it perturbation dataset py. Exactly, the loader is not necessary here. I don't know why. Okay, so we have perturbation dataset in a separate file as well, and then the virtual cell as well. Let's actually call it virtual cell, not virtual cell model. NumPy, are we using NumPy here though? Okay, so we have virtual cell here. So now we are missing those things, but that's okay. That's okay, because now we can also turn it into a package. And now I need to copy and paste a piece of configuration that I always just copy and paste. Let's call it virtual cell perfect. Requirements, we probably will be wise to have requirements. We will have let's populate this with all of our packages. Perfect. And now we can install it. So we'll do pip install our virtual cell. Now, once we have this installed, we can yes, we had it in source exactly. So now we can basically just do from ML utils import gene embeddings, get gene embeddings from ML perturbation dataset and ML virtual cell import virtual cell. Yeah, we need to rename our virtual cell here. And this is now better. This looks better. Yeah, this looks way better. I would probably also extract this later, but for now, since this is a train script anyway, this is perfect. This I would also put into a separate config file. But again, let's leave it right now as it is because it's already usable, I think. Let's just run the training again, just to make sure that it's running. Beyond the cleanup, there is actually cleanup is completely secondary. I just don't want like working in too much of a mess because there are way more urgent and important changes that we need to do here that will actually make it work better or just even work at all. Because right now it works, it works, but it won't be great. Okay, so we have NumPy dependency somewhere. Let's just see where it is. So you needed NumPy after all. Okay. Loading data. Mind you, we are already using cached embeddings, right? So we have the embeddings cached here. So loading embeddings takes absolutely no time. That was the point. Let's wait for it to start to train. Yeah, it's training. Perfect. Great. So now we can stop this. And now we can try to use it. In order to use it, let's go to our playground, of course. And now let's just add, let's just import everything that we need. We basically need this and this. We also want to load the data, load the embeddings. This is all necessary. And we'll also need to import ScanPy. That's it for now. So let's, why would you say so? Because I didn't restart yet. Okay, so we've imported all of our necessary libraries. We are now loading the data and loading the gene embeddings. We've moved directories, so we need to update the root directory because we are inside the notebooks fault directory and not here. Okay, loading data, loading gene embeddings, great. So we have everything in place. And now we can just load our model. checkpoint is torch load. Yeah, close. But not really. This is our checkpoint. Loading works. And now we have model configuration, which is under, I believe, model config. Model config and model state dict. So it's model config and model state dict. And now we basically just and load state dict. And now this should be all keys matched successfully. So now the model, we have as an input, we have 19,360 features. Why? Because it's 18,080 control expression plus gene embeddings. And from the first layer, we have 512 outputs, and then into the second layer goes in the output of the previous layer, and also the final layer is also of the same dimensionality as the control expression that was going in. So yeah, our model works. So now let's, I mean, not works, but it's saved successfully. Let's just, put it in eval mode just to make sure. Just to be sure. Now let's pick a gene. Let's say that we'll knock out TP53. And now what do we need to knock it out? We need control expression, some control expression. So let's just take the control expression. This we could have loaded earlier, so let's do it here. So we have control expression. So we will have sample control expression, and it's just some random sample drawn from the control samples. Okay, and what else? We also need gene embeddings. Embedding, which is, just we are using our mapping that we have here, which maps the gene symbol to gene embedding. Now when we run it, we have the input pair of sample expression, control expression, and gene embedding. Let's just see that there is anything in there. There is. Perfect. This is normalized, so everything is correct. Now we can just basically run the model. So model eval, we already did that. So we need to convert those to tensors, and then we basically convert the sample expression into a tensor, gene embedding into a tensor, and then we pass the control expression tensor and the gene embedding tensor, right? This is our expected input. And we pass that to the model and get the predicted expression. Yep, that's it. So we just predicted expression for the knockout of TP53 based on some control sample. And mind you, we didn't have TP53 we didn't have TP53 in the knockouts, in the training or even validation knockouts. So this is like completely out of distribution. So this is basically just to say that we have a working, exactly as we described it at the beginning. We have some kind of device that works exactly as we wanted. So it's accepts, at least at the surface, right? I'm not saying that those numbers are correct. In particular, especially since this is negative and there is no such thing as negative expression. But keeping that aside for a second, we have a model trained on some historical data that takes as an input some expression that is like starting point, takes a gene to perturb. We represent it as an embedding, but it's a mapping, right? So it takes a gene to perturb and predicts what would happen to this particular cell if we knocked out this particular gene. Now, at this point, we've pushed through the pipe end to end, and we could theoretically end here. But this moment is actually pretty terrible right now. So now let's improve it so that we actually have something useful. But first, let's address let's update a couple of things in this script. There will be more like technical things and general things unrelated to virtual cells. So I will go through them pretty quickly without too much explanation. And then we will again dive a bit deeper into the domain-specific improvements. So let's start with not computing on CPU. Okay, so if we have a GPU available in one form or another, we're going to use it. If not, we are going to use CPU. If you don't have GPU lying around, or if you don't have M-series MacBook, that's fine, it will still work like it used to. Now we just need to move this to the device. We need to move the tensors to the device and those as well. Okay, perfect. So now we basically just said that all of the computations should be done on the GPU. What else? The next thing is, this model is now, this script is now kind of undeterministic in the sense that the split will be different every time because we're relying on set. And this has like an internal seed. So every run we would have a different train validation split. So this would be not reproducible. Now it will be. Do we have any other sets here? Okay. What else? Okay, now this reminds me that actually in the perturbation dataset, exactly, we had this. Okay, I said I won't be explaining things. This is fine. This is the code for randomly sampling the control sample from which we are predicting the perturbation. But this is done with NumPy, and it will work in the current setup because we are not using more than one worker. But if we bump the number of workers to this to have like parallel processing, then since this is not relying on Torch, this would probably end up with all of the workers having the same random seed. So all of the parallel workers would get the same random control samples to predict from, and we don't want that. So we actually want to use Torch. Yeah. Okay. So instead of, now we can delete this. I should have actually done this from the start, but we have it over now. Okay. What else? Let's add weight decay. So to put a little harness on the model. And let's also use scheduler, but not cosine annealing. Let's use reduce learning rate on plateau. Okay, so now whenever we have 3 consecutive epochs without exceeding the best validation, the learning rate will reduce by half. This is great. And now we also need to make a step. Okay, and let's also print the learning rate. Okay, so we see how it changes. What else? We will, let's also, on the same note, let's add overall patience. Now we were, whatever. Now we had, before we had 10 epochs over which we were training. Now let's have whatever number of epochs, because we won't reach those number of epochs anyway, because we'll have overall patience. And if we don't improve, if our model doesn't improve over 8 consecutive epochs on the best validation score, we will just stop training. So now we just need to add best validation loss and patience counter. Exactly. So this will record the best loss, and this will record. Okay, Copilot is suggesting something. Let's see. Okay, if in a given epoch the validation loss is better than the best validation loss, then we overwrite the best validation loss, we zero the patience counter, and we save our model. Great. In other case, we increase the patience counter, so our impatience, and if the patience counter exceeds the overall possible patience, our overall patience, then we stop the model. Okay, and this now I think is better. Okay, so with the technical things out of the way, let me now illustrate what's wrong with this model. Here, remember, we are predicting what's going to be the expression of a given selected, sampled, randomly sampled control cell when we knock down this particular gene, and this is the result. Let's change the gene. And please look at the expression. Nothing moves, right? But we are only looking at 6 numbers, and there's 18,000 numbers. So let's actually maybe look at something that we actually care about. And at the beginning, we even said that we want to see what's the impact of a given perturbation in a given cell. So the impact is the difference actually between the control and perturbed expression. So we need to measure the difference. And now, exactly, let's select top genes according to this difference, but according to the absolute value. And yeah. So now, yeah, we can print top genes. Okay, top 10 predicted changes for gene. Okay. Exactly. Okay, so now for knocking down BRCA1, we can see that those genes were affected the most. So this gene had an increase in expression this large, and this had a decrease, and this had an increase, and so on. So these are the top 10 affected genes. Now, when we run it multiple times, and as you can see, this ranking changes, but it probably mostly changes, like it only changes because we are randomly sampling the control sample. So let's fix this, and now this shouldn't move. Or maybe we can even fix it to something better, which will be exactly control mean. And now we are basically saying how much does a given perturbation make the cell deviate from an average normal cell like this. So now let's change the gene, TP53. Did you see anything move? Okay, I don't know if you noticed, but nothing moves, right? basically the model completely ignores the perturbation that we are giving it. This is called a collapse. This model collapsed on us. So it basically predicts some mean value and completely ignores the perturbation that we are giving it. So that's why I said it's pretty terrible. Actually, it's completely useless. So now let's make the necessary improvements to make it at least slightly useful. Okay, so now let's actually improve the model. I have all of the improvements prepared on the side, and I will be just copying the relevant code one by one, not to dump everything on you at once. We will be introducing the improvements in no particular order, but they all cumulatively do the work. So let's start. Let's start here. Let's start by being more reasonable. And now we are predicting 18,000 numbers. Most of those numbers probably won't even move. For many of those numbers in the data, expression, for many of the expression data, we probably have no change, no variation at all in the training data. So a very common thing to do is just to pick a subset of most highly variable genes. And this is just what we're going to do here. Okay, we are taking training and control indexes. And from those training and control samples, we are picking, there is of course a function built for that. We are picking top 2000 most variable genes, so genes with the highest variance in the training data. We are doing it on the training data, not on the whole set, just to not have any data leakage, so signal leaking from validation set here. This is just to be always extra cautious. And here we have the highly variable gene list, and here we just filter the data according to those highly variable genes. So this is move number one. Move number two is actually a big one, probably the biggest one here, and is kind of an acknowledgement of the reality that we're in. And we are basically addressing the foundational lie that we've been telling ourselves here. I'll show you the move and I'll be explaining as we go. So here we were taking control samples, training samples, and validation samples. But we were pretending that we had something or the other, if I remember correctly, 200,000 samples. Okay, something like that. But at the very start, I also told you that actually doing something like this is physically impossible, gathering such data, because the act of measurement destroys the cell. So we actually do not have paired control and perturbed samples. We just have a pool of control samples and pool of perturbed samples. And what we were doing so far, it was randomly picking a control sample and then giving the perturbation and this control expression to the model and expecting it to recreate randomly selected perturbed sample. And there's actually a ton of technical variation there in this data. Like at the very least, each cell has a cell cycle, and the expression changes dramatically across the cell cycle of a given single cell type. So depending on the time of the measurement, the expression profile will be completely different. That's why, like, randomly matching control samples and predicted target samples is probably not a good idea, and probably why the model collapsed on us in the first place. So what can we match? Well, the alternative is actually to what we did here in the validation, right? We averaged the control expression, and this is something what we can do here as well. So this is called Pseudobulk, because we don't have bulk data, we have single cell data, but pseudobulk is basically averaging the expression and pretending as if we measured the bulk expression of this population of cells. But when we do this, we will have one control sample and 150 perturbed samples. So now when we split it, remember 120 to 30, we have 120 training samples and 30 validation samples, which is not a lot, right? But this is the reality that we are dealing with, and this we will be able to do something about it actually, because we don't need to do like pseudobulk complete. We can do for instance a pseudobulk for the training for the perturbed samples, so average them, but then feed like mini batched pseudobulk controls, so not averaging all of the control samples, but for instance batches of 100 samples, to show the model at least slight variation in the control samples, and show the model, look, something that looks like this should give you this output, and something that looks like this should also give you the same output. But let's go from one extreme, so just doing random, to the other, having pseudobulk control samples and pseudobulk perturbed samples, and we'll see what it gives us. This will also be good for evaluation. So right now, I created this function already on the side so that I don't, fumble in the video too much. So what do we do here? We take the control samples here. Perfect. And then we calculate the mean of all of the control samples. This will be useful later. And then we also, using this function, calculate the mean for each perturbed gene in the training set and the mean for each perturbed training gene in the validation set. And how do we do that? We feed this function all of the data and a given set of perturbation genes. Here we are feeding training genes, here we are feeding validation genes. And for each gene, we are basically calculating the mean of all of the whole expression in our data set for that given gene. Okay, so in our data set, we select all of the expression profiles for that given perturbation, and we just collapse them to one average expression. And this is exactly what we return. We return a list of perturbations, and a list of expression vectors, but this is just one perturbation, one vector. Okay, with this change, we could also adjust the perturbation dataset, because this now basically does almost nothing, because we're randomly sampling a control sample, but there is only one control, which is the mean of all of the control samples. So we're always randomly selecting the same sample. But it's fine, actually, we can leave it just as a reminder that we can expand this model later by randomly sampling, but not a single sample, but again, passing all of the control samples and, for instance, randomly sampling 100 samples and then taking an average out of those. But I'll leave it for later experimentation. This will now change basically nothing because we are always randomly selecting one sample. What else? Okay, we can tidy this up a bit. We can move this to, say, utils. And here from utils, we can import get pseudobulks. Okay, and this is move number 2. Now, move number 3 is moving to optimizing and measuring something that we actually care about, because here we established that we would care in this instance, for instance, for the order of the differences. right? Which genes are the most affected by a given perturbation? And this is this ranking. So we can optimize for that. Basically, we can ask the model to train to improve this performance across this metric. And also probably it would be a good idea to measure this in the first place. So let's first add the measuring bit, and we can measure it basically using correlation. So we have the control expression, we have the actual expression, and we have the predicted expression. And we want to have the best, to our model to give the best correlation between the actual difference. So here we just subtract control expression from the target expression. This is only data. This is data. These are not predictions. And here we measure the difference that our model predicts. So the predicted expression minus the control expression. Now after pseudobulking, we are just basically subtracting average, right? Average target expression minus average control expression, and average predicted expression minus average control expression. And Pearson correlation, so here we have predicted difference and target difference, and Pearson correlation is basically cosine similarity in mean-centered vectors. So here we are subtracting just the mean of the predicted differences and of the target differences, and then we can basically apply cosine similarity to measure Pearson correlation. Let's just add this as well. Or maybe, okay, let's add this as well. So now we can also start measuring it. And we will measure it in the validation of our model. So after computing the loss, let's just add also validation correlation. So here we are using our function. We are passing the predicted expression, the target expression, and control expression. And we are normalizing the sum, just calculating the average over all of the epochs. And exactly, we will also report this as one of our validation metrics, because this is actually something that we really, really care about here. Probably maybe even more than, at least currently, more than the loss. And since we are measuring the performance on the ranking, we might as well optimize for it. So let's also change our loss function. Let's also change our loss function to something that will be more aligned with what we care about, which is the ranking. Okay, so now our loss function has two parts. It has the L1 loss, which is the mean absolute error, and the cosine distance loss, which aligns the ranking. And we also weigh them somehow. I obviously experimented with those numbers before, but this basically now tells them will tell the model to optimize for the ranking and even optimize for it more than the L1 weight, more than the absolute error. We could obviously remove this or set this just to 0, which is effectively removing it, but then this would probably make the model predict expressions that are completely biologically implausible. And in this way, we focus on this, but still keep the model in check in terms of the total expression. So now this component keeps the model, the expression, in reasonable values. And this part, which is the cosine distance, optimizes makes the model optimize for the ranking on the predicted differences, right? So we are calculating the distance, the difference, the correctness of the ranking on the predicted differences between the targeted control expression. Now we obviously need to use this loss. So we need to use it here and also pass the batch expression, and here as well, okay? So that the model is, the loss function is able to calculate the difference, right? That's why we are passing the control expression as the third parameter as well. And now let's also move this out of the way here. We can even move both of those. Let's add eval function, and I mean file. Let's move that there. Okay, nothing more. And now here we can import eval loss function and Pearson difference correlation. And this is move number 3. Okay, after forcing the model to optimize for what we care about and actually measuring what we care about, let's see if this improvement helps our model beat a simple baseline by adding a simple baseline. And again, let me add the high-level code first. So here, after defining the scheduler and before we start training, we just add a short code that basically calculates some baseline, computes our baseline metrics, and prints them out. Nothing else, nothing fancy. So let's say that the baseline would just be training expression. Okay, average expression of all of the training samples. That's it. Okay, so the intuition is, okay, all of the perturbations have some direction. Okay, and each perturbation in a given cell has some, all of the perturbations have some like common characteristic, and we are just predicting that common perturbation characteristic, whatever it is. It's just some mean there. So here we are just taking the mean of all of the training samples, and this is our mean expression baseline. This is our prediction. Whatever perturbation we give the model, it always predicts this baseline. This baseline always predicts the mean, the average expression of all of the training samples. So it's basically a constant vector all of the time. So now let's, I'll copy this and let's go through this. This will actually should go quite quickly because there is nothing new here. We are just passing the validation loader, the mean expression, which is the baseline, and the device to make the computations on the correct device. So here we set up baseline loss, baseline correlation. For each batch of control expression and target expression in the validation loader, whatever the perturbation is, we always just predict the mean expression. Okay, it's always the same mean expression, and we calculate the loss on this mean expression, and we also use this to calculate the Pearson correlation, and then we return the baseline loss and the baseline correlation. Nothing else. So we just go through all of our samples in the validation data, and for each perturbation we always predict the same number and see how it works, basically. So we can move this out of the way here as well. Yes. So now we've imported this, and now we are reporting what is the baseline correlation and the baseline loss of our model. Now the final modification is to our model itself. Since we've changed a lot here, we've changed the data that we are passing to it, we've changed the objective, it would be fitting to change the model itself as well. And this will actually be not that huge of a change in terms of lines of code, but conceptually very different, because two things. First, we want to predict the difference, not the whole expression profile. But this is just changing it like this, right? So now the net is only forced to predict the difference, and we just return the control expression plus the predicted difference, but the gradients only flow through the difference. But we also give the control expression as an input, which is pointless because now it's a constant vector, right? It's just a constant mean vector of all of the control samples. So we can also skip that. And here is a cleaner version of the same architecture. So still we have an input layer and an output layer, nothing else. These are just things to behave, to make the model behave nice during training. And instead of accepting control expression and the gene to perturb, we just take the gene to perturb, in particular the gene embedding of a given gene that we want to perturb. It's projected to the hidden dimensions, and then out of the hidden dimensions, it's expanded to the expression dimension that it's supposed to predict. But we treat this prediction as only a delta, and the model returns the average control expression plus the predicted delta. Now, the only other thing interesting here that's changed is here, because we want to give model a good place to start the predictions, and the good place to start is at predicting no difference. So we are zeroing the weights and biases of the last layer, so that the model starts with just predicting no difference, just so it returns control expression plus zero difference, and then it will try to improve on this starting point. So this is the model itself, and here we don't need to change anything. We would, since we have 150 samples, let's also reduce the dimensionality to something like 16. And now this should be ready to run. Okay, I've actually found two bugs here, which we need to address. The first one is here, because we said that we are going to use pseudobulks, and we are passing pseudobulks, but for the predicted values and not for control values. We wanted to pass the mean control expression as the reference expression from which we are then perturbing, and not again to randomly sample random control examples. And the second one is in the is here. Remember when I said that we are going to add sorted, which was a good intuition, to sort the sets here, and I was looking for other sets, but I skipped this one, because this actually also treats the gene list as a set, and we actually want to have it as a list, so that this order is always the same regardless of the run. Okay, because otherwise this would be again completely undeterministic. What would be the order of the genes? And then because this would be random, the split would be random, and again we wouldn't have a reproducible result. So now we can run this and see what will be the result. Okay, loading. Let's wait for it to end. And now we have a model and our results. And as we can see, this is the baseline loss and the baseline correlation. And this is how the training of our model went. It was a very short run, especially since our overall patience is 8. The model basically just started running out of patience immediately, because we actually didn't improve on the initial epoch loss. So it just degraded over time. Pearson correlation of the baseline is 0.2, and our best Pearson correlation is 0.14. But we can actually quite easily improve on this model using our baseline, because look, our baseline is predicting the mean expression. And currently, we initialize our net to predict zero difference between the control and predicted. So we tell the model, okay, output the control expression and then trying to improve from the zero difference prediction, because we are setting the weights and biases of the last layer to zeros. So we can actually tell the model to predict, to start with the same predictions as the baseline. So let's just do it. We can basically copy this here. And mean control will be this. So we have mean expression, so mean expression of all of the training samples, and mean expression of all of the control samples. And now we are setting the bias of the last layer. Exactly. Almost. We are setting the bias of the final layer. Instead of setting it to 0, we set it to 0 before, together with weights. But now we're setting the bias to predict the difference between the mean training expression and the mean control expression. So we are effectively starting with mean of the training expression, because the model outputs control expression plus the delta between the training and control expression. So we are effectively outputting the training expression as the initial predictions before the net even starts learning. And then we only are telling the model to improve on that. So now let's run it and see how it turns out. And now it's better. The Pearson correlation is now better than that of the baseline, as well as the loss. Again, after the second epoch, it stopped improving, but we now have an improvement over the baseline model, even though it's very, very small improvement with 120 samples as we are using right now. And this simple architecture, I think this is close to, this is quite satisfying actually. This is okay. And we can leave it at that. Now we can test it, but before we test it, we need to do one more thing, because we are actually reducing the number of genes here. Remember, we are reducing the number of genes to only 2000 highly variable genes, both in terms of the input and the output. So we need to record this as well, together with the model, so that later when we are using it, we actually know which genes to use. So let's add highly variable genes. And now you can run it again. Nothing should change, only that the model the saved model will now have this variable here as well, this list of genes. I already ran it. As you can see, I have another ran model. And so now you can run it, and afterwards we can then test this. And we're going to test this just the same as before in our playground. So let's just add our libraries, import our libraries. Let's load the data. Do we have to change anything here? Yes, because we need to filter the highly variable genes, and they are available. They will be available here. So let's just move it there. And here, let's load the newest model. Let's load the highly variable genes. And yeah, now we need to filter the data according to those highly variable genes. Exactly. And that's it. Okay, so this is our ranking of the most impacted genes by perturbation, by knocking down BRCA1. When we change it to PARP1, it changed a bit. when we change it to crass, it changed slightly, TP53. Again, slightly. As you can see, many of those genes actually repeat, so the change is not that dramatic, but at least we can see that the model is not completely collapsed. And it's probably predicting something similar to the baseline, but possibly a slight improvement over the baseline. Okay, before we conclude, let's also go through a couple of immediate next steps that we can do to improve this model. I have listed them all here as the first cell in this notebook, so that I don't get confused with my own thoughts. So the first thing would be to just, after training and seeing which parameters work, which hyperparameters work, I would just retrain the whole model on the whole data set, not splitting into training and validation. Just use the whole training set without splitting. Fix the number of epochs, completely ignore patience. Here I would also change the scheduler to cosine annealing with the final learning rate the same as was in our training run. And then this would should increase the performance of our model, because it now will see more samples, right? Instead of 120 perturbations, we will now expose it to 150 perturbations. So this is improvement, immediate step number one. Next, I would build a simple app around it, because now we've demonstrated it. Like, as soon as we have something useful, I always like to build a simple app. Not only for me, it will be then easier to explore how it works, even for me, but when I want to share it or exchange ideas with anybody, or just show it to somebody, do you find it useful? What do you think about it? Doing it here is extremely distracting, especially for non-coding people. So I would immediately do a very, very simple, create a very, very simple app. Okay, so maybe to illustrate it, I already did, obviously. So maybe I'll show it to you quickly what I mean by extremely simple app, because I really do mean it. This is a Streamlit app, which I vibe coded with 0-shot prompt. It was a single sentence prompt to, I don't remember which model. And it uses Streamlit, and most of this code basically loads the data and is copied from this notebook. And let me run this. We need to install Streamlit. I already did it, but just so that I don't skip anything in this tutorial. And now when we run this, okay. Okay, now it's loading because it's loading the data. And as you can see, this is a Streamlit library, which is like an extremely fast and easy way to build web apps. As you can see, we are using like 1 line of code from this, 2, 3, 4 lines of code to build an app that does exactly what we need it to do, right? So pick a gene and predict the expression and sort it according to the absolute value of the delta. Exactly. So now, as you can imagine, this will be now way easier conversation with somebody who is non-technical to illustrate what you've built and what it's capable of. That's why I said building an app, a quick and dirty, as I like to call it, quick and dirty app is great just as a conversation point. Next, mini batch pseudobulks. This is also something I would try. I'm not sure if it will improve, but we talked about it here when we went from randomly matching controls and training samples with calculating pseudobulks, so averaging over all of the perturbations and also all control samples. We said that here we could basically pass all of the control samples one by one, not averaged, and then sample like, I don't know, 100 control samples and then average their expression to see if we can squeeze more data, like more information out of this data set and amplify the number of samples. This is the next thing I would test. And speaking of samples, just using more data. In the beginning of this video, I also showed you and told you that there is a ton of data of this type, single-cell RNA sequencing data. And I also showed you that there is like hundreds of millions of samples. I showed you the resources that you can look at. So this is an obvious choice. But on that point, you would also probably need to move the model a bit, because you would definitely need to add more context, because those samples will not be of the same cell type and all of the other conditions. So then you would somehow need to inform the network. Look, okay, here is the control expression, and I would like to predict the perturbed expression, but in this cell type, and in this tissue, or in this particular disease and cell type and tissue. So these are the next things you would probably need to marry together with adding more data. And on the same note, right now we basically completely ignore the expression. Our model takes as an input the embedding, but and predicts the delta, right? This is the embedding and predicts the delta, and we just add it to the constant mean control expression vector. We could use the expression, especially when we add more data with more context, because this expression now will be meaningfully different. The control expression will be meaningfully different. But we'll probably do it through some kind of an encoder. You can either use one of the pre-trained expression encoders, or probably at least start with experimenting with your own. And then the most obvious probably thing, I don't know, all of them are quite obvious, but this is the one that you may feel the strongest urge, because it's probably the most cool one, which is just improving the architecture, right? This is extremely simple, basically as simple as it can get. And there are tons of ways to introduce biases into this network so that it learns in a more clever and domain-specific way. And to find some inspiration, you can look at solutions like State or Excel, which are basically state-of-the-art, or other older foundation models, just to get some ideas, because there are some really clever ideas in those papers, in those solutions. And the last thing I want you to keep in mind is that this is an open problem. We're not talking about something that is already done, and now we are talking about engineering of this, although engineering is a big part, as you can see, but this is an open problem. This is still in research, and there are even papers that show that many of the huge foundation models do not beat simple baselines, even like our simple mean baseline, right? Out of the context of the data they were trained on. Other researchers argue that this is a matter of how we measure the models. Long story short, it's still an open problem. This is a hot debate and active area of research. So I also probably highly encourage you to try to stay on top of it and read on it, because it's really, really exciting stuff that is happening here. Yeah, I think we can conclude now. So our goal was to create a virtual cell in Python from scratch. And I think this is exactly what we did. Our model is extremely simple. It takes an embedding of a perturbed cell, predicts the change in expression, and adds that change to the control expression. And this is the prediction of the perturbation effect. We trained it first on randomly matched control and perturbed cells, but then went on the other extreme and completely pseudo-bulked all of the data. where we average the expression of all cells with the same perturbation to get a more stable signal. We wanted to see, okay, what's the perturbation without all of the differences between single cells. Our model is very small. It barely beats baseline performance, but it is the simplest and possibly even maybe slightly useful version of the idea. And now you can play around it and improve it. And that's it. I hope that now you have a better understanding of what a virtual cell is and how it works. Thanks.